Okay, let's get started. Hello, everyone, and welcome to the final webinar in our 5-webinar series on nurturing city soils for healthy organic vegetables. This webinar will be about understanding laboratory soil tests by Roland Wilhelm of Purdue University. I'm Alice Formiga, and this webinar will last about 45 minutes, and then we'll have 30 minutes for questions. So today, I'm very pleased to welcome Dr. Roland Wilhelm, who is an assistant professor of soil microbiology in the Department of Agronomy at Purdue University. And I'd like also to thank the organizers and sponsors of this series, especially Professor Lori Hoagland of Purdue University, who runs the NIFA OREI-funded Tomato Organic Management and Improvement Project. And co-sponsors include the Carrot Improvement for Organic Agriculture Project, the Indiana Association of Soil and Water Conservation District's Urban Soil Health Program, and Purdue University Extension. So with that, Roland, welcome, and I'm going to hand things over to you and turn off my video.
Thank you so much, Alice. And thank you all for giving me an hour and plus of your time today to let me distill some of what I've been learning about soil health and how we use it to better understand the systems in which we grow, as well as, grow our food better. So I will be hoping to give you a bit of a conceptual understanding of where soil health, the movement, comes from, and sort of putting that in place in terms of where we're at now and where we might be headed. My research at Purdue is focused on soil microbes, but soil health is a multifaceted research topic or science. And a lot of what I've learned about it comes from the Cornell team, the Cornell Soil Health Lab. And so much of the content will reflect that, but there are many tests. And I'm not endorsing their tests in any way, shape, or form throughout this talk. I'm mostly taking, taking things apart and putting it back together for you. So step one, part one will be giving you some ideas of where the sort of the history of soil health. And part two will be diving into more specifically some of the indicators and testing methods that are used. So let's start with the origins of the soil health movement. As any academic, I went onto Google Scholar and I got a sense of how many times people have used the word soil health in different publications over the years. And interestingly, there's evidence of that being used in the 1900s by a gentleman by the name of Henry A. Wallace, who was a student at Iowa State University at that time. And interestingly, he was later appointed as the Secretary of Agriculture in the Roosevelt years. So that's kind of an interesting heritage. But you can see that really picks up later in the 80s and 90s and takes off where we are today. And so why might that be? Well, one of the reasons is we had the Green Revolution in the 50s and 60s, in which the mechanization, the use of synthetic fertilizers, improvements in plant breeding, and essentially the intensification of research into how to grow more crops more yields per acre. What happened next? Well, there were some consequences to this. And of course, one of the consequences is our, we're able to feed a lot more people on the planet. That's a very positive thing. Other sort of side effects were, though, that by excluding other aspects of agroecosystems and other properties of soil, we led to the degradation of a lot of agricultural land, a lot of really valuable resources, natural resources, through compaction, the loss of organic matter, through tillage and the release of that organic matter through respiration, but also through wind erosion and the loss through the waterways, and then also some pretty stark and dramatic challenges with gas pollution, so greenhouse gases, specifically nitrous oxide. And so really the soil health movement is a response to the growing recognition of these challenges faced by let's use our cropland as essentially a hydroponic system where we manage it by adding all the nutrients we need. And instead, why don't we take a more multifaceted view of the different functions that soil performs? And so you see this increase in modern conservation practices like no-till, cover cropping, use of organic fertilizers, as well as this sort of testing apparatus. So sort of a way of tracking changes over time, benchmarking different management practices against each other, which ones fare better for these different types of soil health properties. We've actually reached the point now where soil health has been enshrined in the law and legislation in the state of New York. Back in February, they passed the Soil Health Bill, which defines soil health. And I think depending on who you ask, you can often get very different definitions of what soil health is. People often refer to it as regenerative agriculture, and that has also a long history of practitioners. This bill highlights a couple aspects of soil health, and that is, supporting the production of food, feed, fiber, and fuel, facilitating better water dynamics, so infiltration storage, improving nutrient cycling, providing habitat for diverse organisms, improve resilience, helping offset pollution, so any type of fertilizer pesticide you might use. If your soil can help buffer that and break it down, that's fantastic, as well as carbon sequestration. So a whole range of different attributes of soil are being considered in legislation of soil health. If you're interested in learning more about policy in your state or in your location, you should Google Soil Health Policy Resource Catalog. There's a not-for-profit institute called the Soil Health Institute that'll be the first Google hit with that, and there's a whole list of different resources for you to consider. Okay, so the talk is about soil health assessments, and so really let me give you a background on what this means. Soil health is defined by typically three basic properties of soil: the physical, the chemical, and biological dimensions, let's say, of soil. Each of these have multiple different properties that we're interested in, and how those link to specific ecosystem functions. So, for example, in the physical realm, we look at the density of soil, the bulk density, which is a measure of compaction. Also aggregate stability, which is a measure of good tilth, how well water can infiltrate and how well gases can escape from your soil, the soil structure, in other words. And then chemical, and this is slightly different. I'll actually go into a little bit more detail why soil health assessment is different than nutrient analyses, which are historically and widely used as a tool for improving your growth, your crop yields, and the health of your plants. And then really, I think if you ask many people about what soil health is sort of what's new about soil health, it's this focus on the biological, the ecosystem services provided by soils, the living things in soils, and how they can also cycle nutrients, directly interface with plants, and support their growth in different ways. And so we measure soil health by looking at these different indicators. We make these measurements, and we target them specifically because they represent important functions that we're interested in the ecosystem, in our growing lands. Oh, and also they're sensitive to change. Let me take one step back. So it's that they correspond with soil health functions and are sensitive to environmental change, because there's no use in measuring something that takes 5 to 10 years for the dial to shift. So you want to be able to have an indicator that is responsive to different management practices. If you change something, you want to be able to see what the impact of that was earlier rather than late. Now, one of the key backstories about soil health is that when we think of the frameworks, the soil health testing labs, the commercialization of this monitoring apparatus, it's that the typical soil that's being considered for these testing is really a mineral soil in these croplands, these row crops that spread across large tracts of land. They're not necessarily your urban garden or your small mixed vegetable farmer or small producer. And why do I say that? Well, there are assumptions baked into soil health tests. And one of the key assumptions is that you're working with primarily mineral soil. So that we define a mineral soil as anything less than 20% organic matter. And so you can see on the left here, there's a scale, which is a score that's used in soil health tests. And you can see that scale goes up to only 6% organic matter. If you're above 6%, your soil is healthy by this metric. But of course, if you're working with compost or topsoil or really rich peat, your organic matter is going to be sometimes upwards of 40 to 50% of that total mass of your soil, which means that some of these tests don't really always apply to every soil type. people are innovating for different types of soils, and I'll get into that as well. But by and large, I want you all to know, to recognize that much of the idea and concepts behind soil health originate from a certain type of farming practices, a certain type of landscape in which soils have become historically degraded. And so the aim here is to improve those soils with this testing. And here's, again, this is an example of the soil organic matter content in over 14,000 different field sites sampled by this researcher, Nunez et al. And you can see that the average is less than 2%. So this, again, is sort of the backdrop for what is soil health. And I guess the other point is that in certain cases, you might have too much of a good thing, right? You can imagine that when you have too much organic matter, say your rich compost, there's not a lot of, it becomes anaerobic. There's not a lot of oxygen, gas diffusion through that type of soil. So really, if you were looking at an organic soil, it might be the opposite. You might see better health with lower organic matter content, down closer to 20%. I'm not saying that we haven't done any research into this, but I think that probably stands to reason. Okay. And so when we look at these health tests, these plots here are from the Cornell assessment of soil health. And it's the idea that there are also assumptions built into what is good and what is bad. So for some properties of your soil, more is better. Some properties, less is better. And then also for others, there's a balance that you're looking for. It's not add more or reduce things. There's sort of a optimum point. So examples of things where more is better: soil organic matter, aggregate stability, water capacity, and mineralizable nitrogen. And I'll talk about exactly what these things are in the second half of the talk. Less is better. Well, you want less hardness. You don't want your soils to be compacted you want places where the roots can penetrate and grow. You want less bulk density. You want somewhat fluffier soils so that, again, root growth. And then you want fewer pathogens in your soil. And then cases in which balance is better. Well, pH is one example of that. Phosphorus is another. If you add too much phosphorus, the runoff can become a source of pollution in your area. So you want to find that sweet spot so you're not overdoing it and creating pollution. And again, always to complicate things, but this is kind of important when you're thinking about soil health because it has to relate directly to your needs. Probably one of the better ways to think about soil health is not what it's going to tell you to do, but how you can use it as a tool to better understand your system. So, for example, if you're growing plants that have a pH optimum around 7, that's great. But if you're growing bog plants, things where they have evolved to grow in low pH soils, acidic soils, your assumption about what's the optimum health for your soil pH is going to differ, right? So some of this really depends on what you're doing, what your needs are with soil. Another really key aspect of soil health is that we know that the measures differ based on the texture of your soil. So if you have a sandier soil, the ratings, you're only going to be able to add so much organic matter to a sandy soil because the particles, the conditions in that soil do not allow for you to pack it full of carbon. So a sandy soil that has maybe 2% carbon is actually probably a really healthy sandy soil, versus if you had 2% carbon in a really clay loam soil, that could be on the middle of the soil health spectrum or on the low side. And so we often are having to normalize our measures of soil health to different characteristics of our soil. So I think that's kind of the key thing here. And that means that we use these things called scoring functions. And so that's taking the raw value of, say, 2% organic matter, and that's transforming it to reflect, okay, what's the soil texture in the soil? Okay, let's adjust for that. And then we project that onto this sort of S-shaped curve you see here. And that's where we develop our sort of recommendation from. You have low health, low-ish health, and along these different color gradations. So just as a bit of a recap, we have the raw values from these different chemical, biological, and physical indicators that I'm going to introduce later in my talk. Then you have these normalized scoring functions that sort of provide you with a health rating. And then in many cases, the health tests will aggregate those scores and give you an overall health score. And that's what this looks like from the Cornell Soil Health Lab. they'll measure the texture and adjust all of the actual raw values, which is this value column here, and they'll give you a rating, and they'll let you know what are some potential outcomes or impacts of having a low rating of, say, surface hardness. There'll be issues with plants establishing healthy roots, aggregate stability. You may have issues with runoff, and similar things. And in this case, a biological indicator came up as low, and that's this idea of active carbon, which is a measure of available carbon for supporting microbial activity in your soil. So this is what an actual health test looks like. And that's currently, but in the future, it's really quite an active topic of research. There's still open debate about what are the best philosophies behind soil health. Do we want to make it so comprehensive that tests become prohibitively expensive? Or do we want to pick 3 or 4 different indicators that serve multiple purposes and can be measured for $10 to $15, which then allows us to scale up and do more with those fewer measurements? There are examples like this here in which soils are being collected from across the continent and being sort of partitioned into different regions using mathematical models to say, okay, these under this mean annual temperature and this mean annual precipitation and this soil class, this is sort of the curve for what's good and what's bad for soil organic carbon. So refining these transformations into a health rating. And I think that's a really positive move that's going to give people a lot more relevant and meaningful information from where they are. So to summarize, the soil health movement began as really a response to the deterioration of mineral soils in large acreage farms. Soil health is defined by the capacity of soils to provide multiple chemical, physical, and biological functions. And then soil health is monitored by these indicators, and so not exactitudes. Again, one person's top-notch soil health, healthy soil, may not actually serve the purpose of somebody else, right? You're not going to grow peatland bog plants in a mineral soil with high quality soil health. So again, these are tools for you. They're not necessarily prescriptive. So indicators are context specific, but work is ongoing to improve those interpretations. And that's really where the field is headed. We're trying to make more concrete recommendations. We're trying to refine and customize information for specific growers with specific needs. One of the things that generally you can't go wrong with is you want to have some data. I asked Alice, had she ever done a soil health test in her career, and she had said yes when she was starting a new garden. And that's a good example of a good time to do one of these tests. When you're starting something out, having a benchmark for here, this is where we started, and then maybe revisiting it 2, 3, 5 years down the line to see what's changed and how is that relevant. So, and what also really helps with soil health data is that you can share it and you can compare it. I think that's a real value to this. Often people will come to me and say, what does this mean? I've got these different indicators, we've got these different recommendations, but sometimes that's not satisfying enough. And I think one of the challenges is that you really want to compare your operation with somebody else's and see, are they getting better health scores than me? That can really help inform more practical recommendations. So if somebody's using this other technique, they're applying this kind of fertilizer, or at this time they're rotating the crops in this way, that can actually be really useful information when you compare with your colleagues and your peers. So again, soil health test, it's not going to give you fully prescriptive. We're trying to give concrete recommendations, but it's never going to really meet all those needs. But it certainly is a data point for you to hold carefully, take it to an extension outreach workshop and show it to people and say, what do you think about this, these results? I'll maybe hold questions to the end of this talk. Previously, when I gave this, I opened up questions now. But let me just segue into part 2 of the talk, which is going through some actual actual measurements of soil health. On the left of this slide is what we call a rain infiltrometer. Infiltrometer. And it's essentially this giant tank that that creates artificial raindrops, and that will be essentially your soils will be put on sieves underneath this artificial rain, and whatever sort of gets eroded from your soil will be a measure of the stability of your aggregates. So this is one of the tools used by the Cornell team, which is really quite remarkable. Okay, so initially, soil testing really was just a consideration of the nutrient assessment. And this is vital. This kind of information is absolutely, if you ask me, do I get a soil health test, or I have no information about the nutrients in my soil, do I want a soil health test? I would say probably start with the soil nutrient assessment. This is sort of key information about the macro elements that plants need to grow, the pH of your soil, the buffering capacity, things that have very dramatic impacts on growing. And this will also give you very specific information on nutrient management. This is what informs a lot of agronomic practices. However, it is not a soil health assessment. It's not this holistic view of what a soil is and what a soil can do for you. So I don't think it'll necessarily give you all the answers, but soil health is a very comprehensive, valuable, holistic way of looking at your soil. So the Cornell soil health test will give you 12 different properties in total, for each of them separated into 4 different chemical, physical, and biological things. And there are also other tests you can tack on. And so the physical and the biological properties are not what you get from a typical nutrient analysis lab. And so here's just an overview of some of the costs for soil health testing. And the kinds of soil health indicators and tests differ from each group. There's no standard set of properties that are measured. So that's important. So when you're looking at the different costs, take into consideration that different organizations are providing more different tests. So the Cornell soil health test is $90, but that's because they do 12 different tests. And then these nutrient analysis labs, it's often kind of off the menu thing. You can ask for additional tests, and so these are the labs that currently offer soil health testing on a commercial basis. The University of Missouri is the closest one to Purdue University. So if you're from Indiana, that's the place to go. And this is, I guess, let me start with the biological indicators of soil health, which are near and dear to my heart. This is a picture of a bacteria that I isolated from soil and characterized. And soil biology is actually one of the things I want to, make sure you leave this talk with. Is that it's a very new field still. There's a lot left, a lot of mystery left. And that's because we haven't studied some of the microorganisms in soil, because we've never really found a practical need to. They haven't been necessarily, infecting us or our livestock or our plants. There's just a cornucopia of natural history and natural capital in these soils. And so this is a cute little bacteria called Parabricholderia elongata. So there are 3 categories of biological soil health that we really focus on. One is biological activity, so how active these organisms are in performing chemical reactions, transforming organic matter and recycling it back into elements and nutrients that can be used by plants and other organisms. We look at community composition. So these are indicators of who's there, not necessarily what they're doing, but organisms will often reflect a very specific niche that they occupy. So if you find me, you're probably going to find me next to some chocolate, maybe some coffee. And so if you see me there, I might be an indicator of access to snack foods. So that's kind of why we use composition. And these include phospholipid fatty acids, which is a fairly expensive way of profiling communities, but it's quantitative. Whereas microbiome DNA sequencing, which more and more labs are offering, it is I think you can get that at a lower cost, but it's not a quantitative approach. It has its own biases, but it is also a very rich information space that I do a lot of my research with. Community composition can also target root pathogens, so soil-borne organisms that are pathogens to plants. And then the third category of biological soil health, it's a little counterintuitive, but it's the idea that the resources themselves in the soil should be considered as part of that biological health. That there's nothing going to be active in your soil if there are no resources there. And so one of the things that we can manage in our soil to help improve biological health are these resources. So that's why they're part of these soil health tests. And that includes what's called water extractable carbon and nitrogen. So these are sugars and other proteins that are somewhat available to organisms, ACE protein and active carbon, similar types of measures. So let me start by just going into detail about those resources. Water extractable carbon and nitrogen, the largest, we're made up of primarily carbon and nitrogen. There's also phosphorus and other elements like sulfur and iron. But these two are critical for generating biomass. And so there's one of the more popular tests called the Haney test. And that's a combination of a measure of this water extractable carbon and nitrogen. The water extractable part means that this stuff is easy to come out. It washes off quite easily, which means it's a proxy for how available it is to different organisms. So when it rains, how available are these nutrients to things that are growing in your soil? There's the autoclaved citrate extractable protein, which is sort of a measure of the total protein in your soil, but also serves as a proxy for the amount of organic matter, because organic matter is made up largely by protein. These two properties are related. So it sort of gives you not just an estimate of the carbon in your soil, but a total organic matter, how much decomposing material is there, how much biomass is there in your soil. And then similarly, active carbon is a measure of the readily accessible carbon. So we break carbon in your soil into three different categories, generalized categories. One is passive, so that carbon can stay in your soil for many hundreds of years. It's inaccessible. It's somehow become no longer a resource or a meal for things that are living there. Then you have the slow turning over pool, so the things that for a variety of reasons may be less easily degraded over time. And then you have the active pool, and that's what the active carbon and the ACE protein often will give you a good measure of, which is why they're indicators of soil health, because management, when we change something, how we manage our growing area, that can have an immediate impact on this sort of actively cycled pool. And so that's why active carbon and ACE protein are very common measures of soil health. And I actually would recommend those as well. So what active carbon is, just to give a little bit more context, it's this idea, it's kind of a cool test. It's where you add this oxidizing agent to your soil. And then depending on how much of that purple permanganate solution is lost, which it corresponds to how much of that organic matter was oxidized. So when that oxidation reaction happens, the purple color disappears. So the lighter the tube, the more actively oxidizable carbon is present, and that's what this active carbon or POX-C measure gets at. Another really popular tool is soil respiration. So you and I and most living organisms on the planet will use oxygen and convert some form of sugar or organic compound into carbon dioxide. And so if we take a bulk measure of the amount of carbon dioxide that's coming out of a soil, that gives us a nice estimate of how active things are in that soil. So it's a bulk measurement of, and this is just an example. This plot down here just shows if you add no carbon to soil, you have a really low respiration, and really the dose response curve based on how much carbon you add to that soil gives you a really strong measure of how active those soil organisms are. Another really important one is nitrogen mineralization potential. And that's this idea that a lot of the fertilizer we add to soil is very soluble, it's very accessible to plants, but that causes a lot of issues. So when you add nitrogen to soil, it can be a really, like I mentioned before with nitrous oxide, it can be oxidized into gas. It's a potent greenhouse gas. And also that means that it's not getting to plants. So when you add really soluble, easily accessible nitrogen, it can be quickly taken out of the picture. Whereas nitrogen that's stored in organic matter, it's a much slower release time. So the timing between when a plant needs that nitrogen and when it's being released is actually can be in much closer lockstep. So one of the key dimensions of soil health that people are starting to really pick up on is how can we affect this mineralization potential? How can we improve the amount of potentially mineralizable nitrogen in our soils, and how can we make that occur across the growing season and be released at the times when plants need it the most, not just when we have the chance to run our equipment through the field. And then also enzyme assays. These tend to be more expensive and less common because there are also some really some challenges with different types of soil. The enzyme assay can be very biased, but they're also a very direct measure of the types of activity that a soil has. And so plants can't actually generate their own phosphorus. They're not able to convert phosphorus that's stored in minerals and in the rocks in your soil into phosphate that it can incorporate into its biomass. So it actually depends on organisms to perform that enzyme activity to then uptake that phosphorus. So these are other approaches, less common, but soil respiration, if I had to say, pick one, I would say that's probably a really useful tool for measuring the health of your soil and benchmarking and seeing how it changes over time. So this is a sidebar where you have this Haney test. Again, this is a company called Solvita. I'm not sponsored by them. I don't actually know anyone at the company, but they offer a very simple test. It's a colorimetric test where you incubate your soil in a jar. Based on the change in that measuring, you can essentially have to come up with some estimate of how much respiration, how much carbon was respired from your soil. So there are easy off the shelf measures for you to do this at home as well, I think is the point of this slide. And there's also, I encourage you to reach out to me afterwards if you're really interested in measuring some of these things, because I actually looked on Amazon and there's enough materials that you can source from the internet and from commercial suppliers that you can actually develop some of these tests at home. It really depends on how much work you're willing to put into it. Okay, so the phospholipid fatty acids, I had mentioned they're an absolute abundance measure because it's a quantitative approach. We're actually essentially dissolving all of the lipids from cells, the cell membranes, and then we're capturing that and quantifying it. And it can be very useful in comparing the fungi and bacteria in your soil. It gives you information on both of those, and that can be really important if you're working with a plant or in a system where mycorrhizae are important. So again, I think phospholipids have a place, but they're quite expensive, and so they're not as commonly used anymore. DNA sequencing has become more of the de facto go-to. It's cheaper. You can determine the relative abundance of diversity in soil fungi and bacteria as well. That's a little more involved. Typically, when you're doing DNA sequencing, you're targeting one or the other, but you can do both with a little more cost, I would think. And then this root pathogen assay. And so the root pathogen assay is really quite easy for you to do. You can grow a bean. I forget what kind of bean it is, but it's really straightforward. You just take your soil, grow a bean in it, and then you have this scoring system where you look at how healthy the roots are, how branched they are, how blackened they are. Fusarium will cause a sort of blackening, rotting phenotype, and so you can actually just grade it yourself and say, okay, my roots look white and healthy, and that should be that's my soil is doing well. And so this is some work that I've been doing with soil microbes. I just wanted to give it a little bit of a spotlight because microorganisms can be really useful indicators of soil health. And so these are 3 different bacterial groups that are found in soil. And you can see this is using this Cornell assessment of soil health. So you have soils that have low soil health, and the relative abundance of these groups actually tracks with that health rating pretty well. And so I was able to show in my research that the soil microbiome can actually be predictive of the health of your soil to some degree. It's not perfect, but there's enough information based on who's there and what they like to do that you can actually separate out these different health categories. So for example, this middle group here, the Nitrososphaera, they're commonly correlated with conventional agricultural practices, and that's because they're ammonia oxidizers. And so when you add a lot of nitrogen to a field, you actually stimulate their growth, and that's why you have them at elevated abundances in soils that are typically considered poor soil health. And same with this Pyranomydaceae, they're more abundant in these lower organic soils these lower soil health rating soils because they're commonly found in organic matter depleted environments. Okay, so that's the biological set of indicators. We'll move into some physical indicators of soil health. And what this is a nice three-dimensional X-ray of roots growing in soil and how they fill these void spaces. And so roots are actually really struggling often to find territory in soil. And so compaction, these physical properties are quite important. So physical indicators of soil health, one of the key ones is soil texture. But the good news about that is there's a number of different ways of assessing the texture of your soil by yourself. So that these keys, if you Google guide to texture by feel, there's a whole key for you to use the feeling of the soil to sort of identify the texture of your soil. There's this app called Land PKS, which is based on some historic soil survey data, which might tell you about the texture of your soil. There are phone apps like this Slake test, which where you just take some crumbs from your soil, put them in water, and have that camera take a video of the dissolution of that soil particle. And it will give you an estimated measure of your aggregate stability. So there's some really DIY types of tools that can give you practical and very useful information about the physical status of your soil. And this is the rainfall simulator at Cornell. So it's a beautiful big bucket of rain. And I guess another version of that aggregate stability is this so-called dunking method, where you put your soils in a jar in some sort of mesh, and then you can weigh it before and after you've done the dunk. And poor quality soil has poor physical strength that doesn't adhere to itself, that doesn't have a strong aggregate stability. Most of that will erode and sink from your soil. So that gives you a measure of the stability of your aggregates. And what's important about soil texture and soil carbon is that it affects the wetting properties of your soil, how much water a soil can retain. And so this is another pretty key measure of physical health that a place like Cornell will provide. And so that's how much water your soil can actually store. And that differs based on, again, the organic matter quality and the texture, as well as the aggregate structure, porous bulk density of your soil. So recommendations. This is interesting. Yeah, I'm not sure what the recommendations are. I think this is me to recommend to you. So the different types of tests that I just outlined often will show the same thing. So you don't have to do them all. They're not necessarily something that you want you'll need to do. Often they are consistent. So in this picture, you can see here that all of the different biological indicators in green up in this upper left, they're all positively correlated, which means if one goes up, often the other will typically go up as well, which means you can pick maybe just one and use that as an indicator to serve for your biological ratings. And a recent study has argued that that's probably the best way forward with soil health testing, maybe to do two at most, but use one to give you a coarse measure, a second measure to sort of validate what you found in the first measure. And what this study recommended, and this is a massive study from across all of the US, Mexico, and Canada, they found that 24-hour respiration and total soil organic carbon are the two key biological indicators for soil health. So to summarize, there are several options for commercial soil health testing. Each have a different suite of tests. Inquire first before you send off your samples to one of these companies. Ask them about your needs, your situation. What's the type of soil you have and the kind of land use, what kind of farming you're doing before you submit it. The company should have some advice for you on what tests to take, whether their test is actually suitable for your soils. Like I said, many of these soil health tests are geared towards large-scale farms that are using mineral soils. The standardization and the cost of testing is improving. We're refining which tests give us the most information, and I think the future is looking brighter and brighter. So that's a good thing. And also trust open science. So if a company is trying to sell you a test, you certainly want to know how well do these indicators match the function of my soil? What's the evidence that your specific test correlates with, the water storage of my soil, or the pathogen risk that my soil has? A lot of microbiome soil testing companies are saying, we'll survey the pathogens in your soil. But I always ask, if you find one, does it mean it's going to be a problem? The fact that it's just there, how common is it found in the environment, right? It does, just because you detect it does not mean it really poses a risk per se. So ask what kind of evidence backs up the importance of their measures. I think that's a critical point. And then there's some additional resources that I can point you to read more about these soil health tests. Really good one is this comprehensive assessment of the soil health manual by Bianca Mobius-Clune, who was the most recent director of the USDA National Natural Resource Conservation Service soil health program, as well as this soil health analysis set. There's a set of 2 books there. So I did want to leave off have 4 or 5 minutes. I wanted to give you some practical advice also about sampling. So let's imagine you've heard my talk and you're still pretty keen on doing a soil health test. What kind of things do you want to consider before you go out and collect your samples? For starters, you want to ask yourself about your goals. Is it to monitor change over time? Are you about to embark on some sort of new project in which you think your new management practice or the type of crop you're going to grow is going to impact soil health in a positive way or maybe a negative way? Probably it's a good time to do a soil health test so you can benchmark that early stage and then compare it later. Other goals might include looking at differences within your growing area. Say there's a certain plot where you notice your plants often get diseased, or they're not growing as vigorously as in other parts of your field, your operation. Maybe that's an interesting opportunity to compare 2 different soils within your area to say why maybe one's performing worse than the other. And yeah, again, comparing your effective management is always a good approach. So another question you should really ask is, can you sample a reference site in all of these cases? So more information, it's not always easy to say if you compare your sample to somebody else's from 100 miles away, whether those differences are meaningful, but it's much more meaningful if you have a reference site in your area with the same soil type, maybe unmanaged, maybe where you're not growing anything. It could be just sort of left fallow. It could be something where the sidewalk it just ends and there's a little piece of soil you can dig up. So and if you have the opportunity to set aside a parcel of land, so you're embarking on a new way of managing, set something aside and don't touch it. Just leave it as an unmanaged reference plot. That can be really effective as a tool for comparing your changes in soil health. Another really cool project that I love, it's from Malawi. It's called the Mother Baby Trial. So getting involved with researchers in your area. There are often opportunities, extension agents and university researchers or they're not-for-profits like the Soil Health Institute. They're doing larger scale studies. They might be really interested in partnering with you to have you replicate one of their treatments in your plot. And that kind of information is super critical because what researchers have access to are fairly centralized, localized research farms that we have tons of data. It's very well characterized. We know what's happening in this area, but it's not necessarily the real world, right? There's so much variation in the real world. And by partnering with people who are growing things, and that helps us actually account for that variation. So how effective is it in a field trial? It might look like there's a difference, but if we start sending that out to a bunch of different people who are growing things and we see no difference, that kind of information is super valuable. And soil health testing can be a good example of how that might work. So when you're taking samples, remember there are always edge effects. Do not sample super close to any of the borders of where you're growing things. Also, try to composite more than one soil sample. The ideal is at least 5 that you've taken from a random walk through your plot or sort of a random design. And that sort of covers sort of representative differences within your field because there can be a variation within that. Avoid obvious outliers. If you look at your plot and you see, okay, this is always soaking wet, it's probably not going to be very reflective of the average soil health or the average nutrient content of your soil. So do use some of your better judgment. You don't have to do it completely random. You can certainly leave out a part that looks a little different than the rest. So again, I want to hammer this home. It's great to have a reference plot. If you can do two soil health tests, do it, because that'll help you interpret the results a lot better. Okay, this is the last bit of technical information, but when you go out and do a test, you just need a straight shovel, a bucket. It helps to have a GPS, what your phone would do, a cooler, and a Ziploc bag. And so you dig, I'm not actually sure how to describe this, but you dig a hole, and then with one of the banks of that hole, one of the walls, you just take a 2-inch slice, and that's what you'll put in your bag and ship off to the testing group. So it's usually about 2 inches by 6 inches is the ideal size. You bag it and include the information about where it's from, the sampling date, and any information that will help you track this over time, and you send it off to the company. Now, again, in closing, I want to say that soil health testing, it helps one see the inner strengths and weaknesses of a soil, but it's certainly not the only tool you have to improve soil health. It shouldn't necessarily tell you whether or not you should be practicing good soil health promoting practices. So, for example, we know that there are basic principles of improving soil health. If you minimize the disturbance of your growing area, if you maximize biodiversity, crop rotations, intercropping, cover crops, and if you maximize soil cover and maximize living roots, you're more than likely going to be improving the health of your soil. So there are certain practices you can do without having to have a test. Again, but a test will show you the inner strengths and weaknesses. And this is just information from a recent, I went to the Tri-Societies meeting in Baltimore, and we were talking about what are the things that really improve soil health the most. And this, again, is that same study I've been referring to from the Soil Health Institute. And it really, if you look at this, rotational diversity, crop count, the number of crops that you plant in one season, they don't really impact as much as just using organic manures, cover crops, and retaining residue. So in other words, adding organic matter to your soils is a very simple way of improving your soil health. So I think that's a nice takeaway message. It doesn't have to be super complicated. A lot of the question really should be, how can I best achieve these goals? And it helps to have a benchmark like a soil health test, because you can show how far you've gone. But again, it shouldn't. If you can't get a soil health test, don't let it stop you. So just want to point out some leaders in soil health. There's farm groups, there's government programs, there's university extension, there's not-for-profits like the Soil Health Institute, and there are soil health testing labs. Each of these will give you some solid advice on how to test your soil. And again, focus on organizations and people who are transparent and will tell you, this is why we think this measure is good for you. And so at that, I'd like to just stop and ask for some questions.
Okay, thank you very much, Roland. That was great. I really appreciated the advice about how you might not need every single soil test that's available. I thought that was really practical. So, we're going to move on to our question and answer period. We have a lot of questions coming in. We'll be answering questions for the next half an hour or so. And I just wanted to remind anybody who might have come on a little late that we did record this, and the recording will be available along with the other webinars in this series about nurturing city soils for healthy organic vegetables on the eOrganic YouTube channel, and also the eOrganic website at eOrganic.org. I'm going to just open up the Q&A box, and we'll just answer as many as we can. Okay, we had a question about whether or not you're familiar with the microbiometer. And when I saw that, I had to look that up, and it's a device that's sold to measure, I believe, biological activity in soil.
Yeah, I have seen it. I think it's a sort of measure of biomass or biological activity. I remember looking at that and thinking it was overly simplistic. One of the challenges is that the type of activity you measure will be very dependent on the soil texture and soil type. So if that company is not including that in their score, if they're saying I can give you any estimate of your biological activity with just this simple colorimetric assay, I would be deeply skeptical.
Oh, by the way, I just wanted to mention that on the eOrganic YouTube channel, there are a couple of videos that were made by Esther Thompson of Utah State University that have sort of demonstrate how to do several simple soil tests that you can do at home, various things with soil texture. And then also she also demonstrated that Solvita test that you mentioned. So I just wanted to let people know that if you just kind of want to see how you can do that at home, she did a demonstration there a few years ago. Okay, so let's see. Have the mycorrhizae been categorized? And if so, are there specific groups for specific plants? And where can I find a list of these fungi?
Yeah, they have been. I think that's one of the most active topics of research right now. It's a really hot topic to better understand which mycorrhizae are sort of critical for plant development. So one of the more interesting results is that people are selling these mycorrhizal inoculants, and there's some really good evidence that if you grow plants in a microbe-free condition and you compare them to when you add in the mycorrhizae, you see really strong plant growth effects. So it's almost night and day how well the plants do. So it's very clear that they depend on their microbial partners for accessing nutrients and for being healthy. One of the questions becomes, how well are plants able to recruit these organisms themselves? And so often we find when you take that same field trial and you add these mycorrhizal from an inoculant and you put them and you compare them to plants that are grown in the field with natural communities able to access them, they fare almost the same. So this idea of, are there certain mycorrhizae that we really want in our soils? It's probably the case, but can others is there a flexibility in the recruitment of mycorrhizae? Those are still questions we're trying to tackle. For sure, we know that for example, when you're planting trees, certain pine trees, they have commensal, so they'll inoculate mycorrhizae at the very beginning of those to help push forward that process to stimulate the colonization. And so yes, they have been categorized typically based on their taxonomy, by their ancestry, how we define what a species is. So yeah, that information is out there. I don't have a specific resource where you can find that. One of the resources used by academics is called Unite, and also FunGuild. I'll put that in the chat box. This is a really interesting way of seeing assigning different ecological functions to fungi based on their name.
Cool. Okay. Let's see. We had a question about what is active carbon in the soil?
Yes, active carbon. So I think I gave a specific, that the test itself is a measure of the amount of oxidizable material in your soil. What that corresponds with is roughly the type of carbon that's soluble and readily accessible, and often also things that are sort of plant derived. So it's a good proxy for the amount of organic matter that's in your soil.
Okay. Okay, well, hopefully that answers the question here. Let's see.
Actually, sorry, specifically whether it's organic carbon or organic matter, it's any oxidizable material. Sorry, I just noticed that I hadn't been answering that question. It's just a change in that the tint and color of that solution. So any oxidization that happens will reduce that will appear as if it's organic matter, but it correlates quite well. There's some pretty strong correlation between organic matter and epoxy, and organic matter is really tightly correlated with organic carbon too. So.
Okay, great. Let's see. Do you have an idea of how well-versed these different labs are in selecting appropriate tests for soils in different regions? One example I think of is available phosphorus testing. By default, many people will use malic extractions when looking for available nutrients, but that test does not work well in calcareous and alkaline soils like we have in South Florida. So an Olsen test would be more appropriate. So do you think Cornell's staff would be able to select tests based on regional soil needs? Great question.
Yeah, that's a great question. Yeah, I mean, without casting any shade on anybody, I'm not sure. I know that they don't have, I'm not sure if they have that built into their workflow. Whether somebody looks at the region of your soil, like where your soil's coming from, and says, maybe this is the better extraction. It seems to me that the workflow is fairly standardized. And so you might be able to ask them to do something slightly different, but I don't think so. So that's a really good point. So sometimes looking around for different commercial testing facilities closer to home can help. Hopefully, yeah. But great
Yeah, maybe
question.
calling them first to ask for a recommendation specifically for your region and talking with them before just kind of sending off the sample might be helpful as well if there's, if you're concerned about that. Because I think, yeah, I mean, a lot of people aren't aware of all these. I mean, I think most gardeners probably wouldn't be available, or farmers wouldn't be realize that there are different tests for all these that might be most appropriate for different regions. So
Yeah, I think my comment there too is that these groups are getting better because they've gotten a lot of flack about this exact thing. Like how, soils are so diverse and they have such different chemistries and such different textures. How can there be a one size fits all solution to this? And I think they're very open about that. So I don't think they would be offended if you said, hey, can you tell me a bit more how appropriate my test is for this soil?
yeah, it sounds like it's really worth asking. Okay, so another question, is POXC an indicator of soil organic carbon or soil organic matter or both?
Yeah, I think I just addressed that one previously. I may have done both. So it's both, I would think. But again, it's a measure of the total oxidized material. So it's not necessarily even specific to organic matter. We just know that when you repeat this test many, many times, there's a strong correlation with organic matter content and the readings from that oxidizable test. What I like about it too, though, the other thing about soil health tests is they're designed to be somewhat not basic, but they're not super sophisticated tests. We can always do more sophisticated testing if we need be, but these are supposed to be quick and dirty. And I think POC of all the tests that I've found is a pretty responsive and effective soil health indicator. So it's one of the ones that works pretty well, I think.
Okay. Are phospholids tests more informative for farmers who are using regenerative practices that greatly reduce soil disturbance?
Yeah, so I have a mixed feeling on phospholipids. I'm sure you might have noticed. I did a lot of work with phospholipids during my PhD, and I found them very useful for certain types of questions, but they're really expensive and labor intensive to do. So I think the amount of information you gain from that compared to other tests is minimal. So if you think that there's something really important about fungi in your soil, I would say that's a great idea. There are other ways of measuring disturbance, though. So if you're just interested in knowing, is my soil more or less disturbed? I don't think PLFA would be the way to go. Actually, the microbiome is a great indicator of disturbance. In fact, some of the most recent research I've done, it's probably a very reliable indicator of when things are when soils have been disturbed. And that's because we have, just like in a larger ecosystem scale, you can imagine we cut a road through a forest, and along the side of that road there's basically barren soil. Now over time you'll start seeing bushes grow and then larger plants grow, and there's this whole succession that happens, and that happens with the microbial community as well. And it just so happens that we can really pick out those differences. So there are sort of early stage fast grower weedy species, and there are late stage organisms that take a while to grow, whether it's because they're slower growing or they depend on other conditions to arrive. So I think if you're looking at just for disturbance, the microbiome in general, the DNA-based testing would probably be the way to go. It's going to be cheaper. I mean, this is what my lab does, a lot of DNA-based testing. And I don't think gives you all of the information you need, but it does provide more, I think, than PLFAs. I think PLFAs is not the ideal anymore.
Okay, we're getting this question from a couple of different people. How often do you need to test your soil? And can the soil change that fast for some physical and biological indicators?
Great question. again, so part of the decisions behind which tests to use for soil health are trying to get ones that are most responsive, that react quickly. So you notice that soil organic matter is part of the test, but we know that that can take a long time to accrue, right? Soil organic carbon takes decades to build. So that said, how often should you test? I mean, some things, if you're interested in so here's a good example. So there's Pivot Bio, this new company that's selling carbon credits to farmers, and they're expecting the farmer to sort of essentially grow their carbon over time. They do a soil carbon test at the start of the period, and they only do it every 5 years later. So 5 years is probably an okay interval. Again, my recommendation is to do it whenever you think you're trying something new, where you might learn something by having a before and after sample. I think that really is where the strength of these measuring health assessments come through, is based on references. How is this management practice changing the soil compared to this one? Is it improving the active carbon? Because you will see changes in active carbon pretty quickly. If you add, for example, the simplest example, if you add manure to a field, you'll see the active carbon spike, right? And so maybe you want to add that manure and test a year later. Is the impact of my addition of manure fertilizer still having impact on the active carbon? So you wouldn't measure it beforehand. In this case, maybe you just say, here's a plot where I didn't add it. Here's a plot where I did add it. A year later, do I still see a spike in the amount of active carbon? That would give you an idea that, oh yes, this approach is actually helping when it's in terms of feeding the soil activity.
I really liked your suggestion about, maybe several people doing a soil test and then comparing the results with each other. And then you can kind of see, if somebody has really healthy soil, you can look and see what have they been doing in their farm or garden that's made a difference. So yeah, that was a really great suggestion. Okay, so we have a question here from Joshua. He said, great overview on this topic. And he works on soil health for urban sites for the NRCS, the National Resources Conservation Service. One interesting question that has come up is, what are good or appropriate soil health indicators for testing sites where people are growing in constructed raised beds or containers and their soil is essentially a potting soil or manufactured soil, not a mineral soil? Do you have any thoughts on good indicators for potting soils and media?
Yeah, I kind of avoided this question because I don't have a good answer for you. I mean, if you're using one of the tests that previous speakers in this webinar series have hit, is heavy metals are important in urban spaces. So if you're using any soils from around that area, then do a heavy metal test. If you're importing that topsoil from a commercial provider or somewhere where you trust that there's no heavy metals, it's not. I think in those soils, really, one of the challenges is just having good aeration. You really might consider adding perlite and other things that can really help prevent that soil from slumping and becoming anaerobic and difficult for roots to grow in. So I think this is partly why I started with that historical background about soil health, is that soil health testing is only now really starting to tackle urban environments, constructed soils. There's just not as rich a knowledge set. Lori Hoagland here at Purdue is one of the people who's tackling these questions. But again, yeah, I don't have any good advice for you. I'm sorry.
Okay, so Cheryl just commented that the A&L Great Lakes Labs in Fort Wayne, Indiana, is what they use at the Allen County Purdue Extension Office. So she said they've been using them for many years as well.
Do they do soil health testing? They do,
Sure.
I guess.
Yeah.
So
okay, well,
I checked recently and they had only listed nutrient analysis. So my apologies if I have missed them, but I think they do soil testing for sure. Purdue sends most of their soils there as well, but they essentially don't do these newer tests where they're looking at the physical and biological dimensions of the soil.
okay, and then also Caleb commented that Penn State soil testing will test soilless media. So yeah, we learned in previous webinar that there are quite a few labs that will also do tests on different media. So okay. Let's see. Is there a test for arsenic in soils that you can perform at home, or would this be a tissue sampling test?
I'm not familiar with how to measure arsenic. I would think it's very much not something you do at home. No. The home tests for soil health that you can do are the respiration. I know a number of brewers, people who brew beer or other alcohol. A lot of the same equipment can be used to do a soil respiration assay. In fact, I gave a guy a protocol not so long ago. So if you're interested and you have brewing equipment, I can send you information on how to build your own soil respiration analysis kit. You can actually get permanganate, potassium permanganate, also online, so you can do the epoxy test yourself. Some of the stuff is certainly like the Slake's test. Most of those you can probably get away with doing, soil pH for chemical analysis at home. You can get away with doing a respiration assay at home, and probably even your physical aggregate stability tests at home. So a number of these things, I would definitely start there. The difference being that these soil health testing labs, they have a pretty rigorous set of protocols. So you know that they're going to do the same thing day in, day out, and that's the one advantage.
Okay. How can these tests help determine carbon drawdown or sequestration?
Yeah, I mean, they're giving you a measure of soil organic matter. So they'll give you the raw value as well as the untransformed value. The raw value, which is not transformed into their health rating. And so you have that information. So the next year you can go back and you can repeat that test. And if you see it increasing, that's a sign that you're building carbon in your soil. One of the challenges, though, is that most people don't. You find that the carbon measures are variable from year to year. So you want at least to give it more time. I'd say 3 to 5 years before you do that test again, if building carbon is what you want. There's also interesting tools. So if you're actually sort of a small scale farmer or a large scale farmer, there's a USDA tool called Comet, COMET, that you can put in a bunch of information about the type of your soil and sort of the region you're in, and it'll give you a prediction about how much carbon you can sequester in your soil, because there is a cap. Like, it's not something we can continue. Your soil cannot infinitely absorb carbon, right? There's sort of a plateau region, and so this tool will let you know, essentially what that plateau is, and then you can say, okay, when I got my measurement, I'm here, which means I've got a way to go. So maybe approaching this with adding more organic matter or compost or retaining plant residues, that might actually have a big impact.
Okay. Okay. So Steve asked a question: is there a best recommendation for small lot urban gardening?
Yeah, again, for soil health, your soil is super healthy by these standards, right? Most large scale or even small scale farmers would love to have a topsoil potting soil mix, like many people use in their raised beds. And so for small urban farming, for sure, it's a priority to get a heavy metal test, some sort of assessment in that case. But if you're doing it the reason that urban farming and soil health is trickier is that if you have access to a composting program, there's a lot of organic matter that comes into cities, right? And it can be concentrated and you can actually access it pretty easily. Whereas when you're covering large swaths of land, you have to come up with other methods to build that health. So I think in many ways, urban farmers have it a little bit easier with the kinds of tools they have to build soil health. You can get plenty of organic matter to put in your soil.
Okay, and then we had a question from John about how home gardeners can apply these concepts with a limited budget.
That is
Great
the key.
Question.
That is the key. Yeah, and I think that's what most people who are proponents of soil health would, not want you to be caught up in the testing part of things. You can, again, those 4 key. Let me see if I can back it up. There are 4 principles here that the NRCS and a number of organizations promote, and that's maximizing continuous living roots. So roots essentially feed the soil. Plants are putting their carbon into the soil, which really stimulates the roots die back, and that builds organic matter, and it also helps create improved infiltration and all those things. So maximizing those roots, minimizing disturbance, maximizing soil cover, and maximizing biodiversity. Those 4 key components are things you can do without having to do any testing at all.
Yeah, those are really important principles there. Let's see, we have time for a couple more questions here. I'm just kind of going through the list here. There's lots of, Carl very nicely shared a video from Minnesota on lead testing. So thanks for that. Oh yeah, we had a couple questions about whether or not the slides might be made available. I don't know if you would be okay with that. If so, I can then post them along with the recording. If you just Google Nurturing City Soils for Healthy Organic Vegetables, that's the name of this series, you'll be able to find all the recordings of the webinars in this series. I can try to remember to send you all a link to that with the follow-up email for the webinar or the survey, just so that you can access all the recordings. And the recordings will be also available in the same place and on the eOrganic YouTube channel. So I'm just going to make a note here to make sure to send the follow-up email with the link to the recording or the survey. We're going to send a follow-up survey, so that should have a link to the recording as well. Yeah, sometimes I forget, so I just have to make sure I write it down here. Okay, so we have time for just a couple more questions here. So I'm just going to go through, let's see. Do you have any thoughts about using field qualitative tests of soil? Do you know what that means? Okay.
No,
Not sure.
The person can clarify, yeah.
Okay, yeah, if they can clarify that. Okay.
I guess qualitative tests, like, people are now taking cell phone pictures of soil to see if to assess carbon content. I actually would say I've heard a few people speak about that, and it can work. The images, the quality of images we can snap with our phone can be used in interesting ways. So do keep an eye on that. I don't have anything to recommend in that space, but one of the challenges is that soils have different colors to begin with, right? The mineral content, they can be wetter or drier. So just going to the field and snapping a picture and having like all of the information pop up on your phone is a fiction, really, but there will be innovation in that space for sure.
Okay, last question here that we have time for. Are nematodes beneficial to soil health?
Not if they're plant pathogens, but yeah.
Not if they eat your carrots.
Yeah, not if they eat your roots. But yeah, I mean, nematodes, other larger macroinvertebrates, these things are really good indicators that your soil is healthy. I caught a talk on nematodes, but they have to be indicators. Like, you have to know what the ecology of these organisms are to interpret whether or not their presence is a sign of a healthy functioning food web, or whether they're actually there because your plants are now falling victim to their prey. So.
Okay, that's great. Thank you, everybody, for asking all those questions. It was a great discussion. I think a lot of the questions were really useful to a lot of other people that might not have thought of the question, but yeah, it was really helpful. So thank you. Yeah, thanks so much, Roland, too. That was a really informative and, easy, at least I thought so, to kind of access presentation. And thanks for answering all those questions. And like I said, I'll send everybody a link to the recordings and the slides. And thanks a lot for attending this series. For those of you who stuck with us, we don't have any webinars on the schedule right now, but we're going to have a new series this fall about plant breeding. And then we have some other webinars on some organic research projects that are finishing up, and some that have just started actually coming up this winter. So we'll let you know about those. So thanks, Roland, again, and thanks to everyone. Bye.
Bye-bye.